Hello, this is Richard from ioLight. I'd like to tell you about our new version of our compact cell imager. So this new version of the compact cell imager has dual fluorescence wavelengths. So it's bright field, but also blue excitation fluorescence and green excitation fluorescence. So the compact cell imager, as you can see, if I hold it up against my pen here, it's very small. It can go inside an incubator. It's all sealed up, so it can work for days on end inside an incubator, or it can be used in a safety cabinet, or of course on the lab bench. It takes images from this point here, looking up through the bottom of a tea flask, or a well plate, or a slide, or any other container that you want to put on the surface here. The images are sent wirelessly to a tablet outside of the incubator, and it'll do Z-stack imaging as well as time-lapse over several days. So the Z-stack feature is great for thicker samples such as organoids or embryos. So we already have a version of this which is bright field and blue excitation fluorescence, but this new version has bright field, blue excitation and green excitation fluorescence. So I'll just show it you working. So this is a test slide, which is a cross section of a plant stem which has been stained with a couple of fluorescent dyes. So if I put that on in bright field mode. So here we go. You've got focus control on the front here. You've got fine focus on the screen of the tablet, which is obviously outside of the incubator, so you can focus this without opening the incubator. The width of the image here is 1.2 millimeters. So the level of detail that you're seeing is about like a 10x 0.25 NA objective on a standard inverted microscope. You've got a couple of bright field options here on illumination. You've got the standard illumination, and you've got a low numerical aperture illumination, which is good for mammalian cells. There's also a digital phase contrast option and a high contrast option as well. So that's it working in bright field mode. Now I can turn the bright field illuminator off, switch over to blue fluorescence. Here we go, so that's the blue illuminator, and you can see immediately we're getting the green fluorescence from GFP and other dyes in this particular test sample. So here we can see we can move the sample around. It's a perfectly good live image. So this one here, so that's the blue excitation giving green fluorescence. So I can now switch over to green excitation, and now I'm getting the red fluorescence. It's not particularly bright on this particular test sample, but there's the red fluorescence coming out of this particular sample. So there it is, running green fluorescence, excitation fluorescence, and blue fluorescence excitation. So if I move over to some more realistic slides now. So this slide here is a student slide. It was done as part of an MSc, and it's about 2 years old now. So the fluorescence intensity has decayed a great deal, actually. So it's a very good test of the microscope. So these are some mammalian cells that were stained by a student as part of an MSc with GFP. So if I put this on, here we go. Instantly, there we go. We can see the mammalian cells immobilized on that slide. They've been there for a good 2 years. And you can see them fluorescing nicely green. So that shows the blue fluorescence very nicely. For the green excitation fluorescence, if I switch over here, I've got another slide that is a cross-section of a mouse brain that's been stained. So here you can see fabulous red fluorescence there. Very low background. Great signal there from this cross-section of a mammalian of a mouse brain. So there we have it. It's a compact cell imager. It's for mammalian cell imaging from within an incubator or a safety cabinet. It does bright field, it does blue excitation and green excitation fluorescence, it does time-lapse, it does Z-stack. So if you'd like to know more, check out our website at ioLight.co.uk or email info@ioLight.co.uk. Thanks for listening.